Great, thanks William. So I'm very happy to be here today, and I definitely would not consider myself an expert in Altmetrics, so it's kind of interesting to be in this particular session, but I do kind of consider myself an expert in reproducibility, so hopefully I can talk a little bit about how we're considering reproducibility as an important altmetric that should be considered in research assessment. So when William asked me to participate in this talk, I thought about why do we actually have metrics, and I think of myself as much more of an end user, so I'm actually a scientist. I still do research, and I guess I kind of think about why do we actually have metrics as an individual researcher and how we're assessed and think about the impact of our work. So the kind of two main reasons that I came up with were the need to assess the impact of research funding, so how do we allocate the funding to the best researchers, and also how do we think about promoting and hiring of the best researchers. So I think these fundamentally underlie really the basis of a lot of the measurements we make about research contributions. So then I sort of thought about, from my point of view, how do we currently assess impact? And I guess as a researcher, the things that seem to stand out are really around still traditional measures of impact, so journal impact factor, and also more recently article level metrics, so number of citations and number of downloads. And these seem to be really dominating a lot of what we look at for assessment and research funding, and also for promotion decisions. But then I started to wonder, are these metrics actually associated with research quality? So I guess I think about research quality mostly as whether those research results can really hold up and be reproducible when another researcher tries to build upon them. So actually, when you look at the data, it suggests that there really isn't very many correlations with these traditional factors. So no replication studies that have been performed so far have found any correlation with reproducibility and citations or with journal impact factor. So these are from 3 different studies that have come out from the National Institute of NINDS, which is the National Institute of Strokes, I can't remember the exact words, and then 2 pharmaceutical companies, so Bayer and Amgen. So in terms of whether these metrics are actually associated with research quality, our current evidence would actually suggest that they're not. So by measuring impact with metrics that are not associated with research quality, what do we actually promote? And I think when you think about metrics, they're really about incentives and how we're motivating scientists and rewarding them. So they are very important for what we incentivize. So there's now considerable accumulating evidence that many of our research results that are published are not reproducible. So based on those initial studies that I showed you, there's been surveys that have been conducted in a variety of different sectors, but this is one of the most highly cited. This is from MD Anderson Cancer Center, where more than 50% of respondents to the survey talked about their inability to reproduce published data. And many of them found that they could never actually find the underlying cause for this lack of reproducibility or resolve the lack of reproducibility. And frequently they were met with very hostile reactions from the community. These are some of the studies that have come out looking at replications. So these are really the only things that have come out in terms of actually getting some data about reproducibility. So from ALS TDI, they actually looked at the whole ALS field. So they replicated 70 drugs that had been used in 221 independent studies. And they found 0 reproduction of the findings that were published. And some of these led to clinical trials that failed in clinical trial, and their hypothesis was that these failures in clinical trial could have actually been prevented if we had have done replication studies before the clinical trials took place. The NINDS, they then sponsored 12 spinal cord injury model studies, and this was based on their looking at the clinical data and actually seeing that really nothing that was coming out of these preclinical studies was translating into the clinic, so they were all failing in clinical trial. And so they wanted to understand, was it a problem at the earlier phases of research, so were the original studies actually reproducible, or was it that the models just didn't apply in the clinic? And what they found was that indeed it was actually a problem with the preclinical studies because only 2 out of the 12 could be reproduced. And then the two most famous studies are from the pharmaceutical industry, who reported that for them, with their in-house target validation studies, for Bayer, 14 out of 67 studies could be reproduced, and for Amgen, 6 out of 53. And so for both of these pharmaceutical companies, they really talked a lot about how basing their target validation programs or their drug discovery programs on academic research was actually a very costly endeavor for them, because they really couldn't build upon the results that were published. So in terms of measuring impact with metrics not associated with research quality, it's arguable that we're promoting high impact but not high quality research. And when we think about what impact non-reproducible research can actually have, I would argue that it really can't have any impact because it can't advance science. So we think that reproducibility is an incredibly important altmetric that's currently really not measured or incentivized for academic researchers. So how can we measure reproducibility? The way that we measure reproducibility is by verifying key experimental results using independent replication. And this allows us to identify robust reproducible research. One of the interesting findings that came out of the Bayer study was that replications alone didn't just show whether the individual experiment was reproducible, they also were able to identify really robust results. So actually introducing a little bit of variability by taking an experiment and putting it in a new lab provided additional data about the robustness of that experiment. So they stated that either the results are reproducible and then showed transferability into other models, which is actually a very good signal for whether something will translate into humans. Well, they actually were able to identify that, or even a one-to-one reproduction of published experimental procedures failed. So one of the issues with using reproducibility as an altmetric is that there's actually very few reproductions ever conducted. So replication studies are very difficult to incentivize, and this is for a variety of reasons, most notably that they really don't have a lot of impact on a researcher's career. And in addition, they actually are often associated with a lot of hostility from the research community. So it's very difficult to get researchers to publish their failed replications. And that's what we saw with the results that came out of the MD Anderson survey, where less than 30% of the researchers that failed to reproduce a finding ever published that fact. And many of them cited that they were too afraid to publish because they felt that it would have a negative impact on their career. In addition, which we have found to be very true, is that replication studies are very hard to fund. There's a lot of interest from funders in promoting novel exploratory research, but very little interest in confirming those findings. This is actually a quote from Pamela Ronald, who retracted two of her studies that were published in Science and PLOS, and she retracted them because she realized there was a major issue with a reagent that was critical to her results. And for her, one of the things that was most difficult was she actually could not obtain funding to repeat her results that had already been published, so she couldn't actually replicate what she'd done in order to determine really were the results correct, and there was just an issue with this reagent, and she could really never get to the bottom of it, and so she just retracted the studies. In addition, Bruce Alberts, so even at the highest level, has really encouraged funding agencies to consider replications, but currently there's been very few, or to my knowledge, I think none besides the UNINDS, of funding agencies that have actually called for funding of replication studies. So using reproducibility as an altmetric will really require a commitment from multiple stakeholders. So it really needs funders, publishers, and institutions to actually encourage this by promoting replication studies. And we believe that by promoting replication studies, we'll be able to identify and reward high-quality reproducible research. So the first major public replication study that's ever been funded is the Cancer Biology Reproducibility Project. This is a project that I started with William, and we work on this with the Center for Open Science. This is independently replicating the key experimental results from the top 50 cancer biology studies published from 2010 to 2012. And how we defined these was based on traditional metrics of impact. So we looked at cancer biology-related search terms in the Web of Science and Scopus and combined that citation data with number of Mendeley readers and altmetric.com scores. So this allowed us to really combine an impact score that we felt represented many of the different factors that are looked at research impact at the moment. We then excluded reviews, clinical trials, case studies, and sequencing papers, because many of the sequencing papers actually don't have the same level of reproducibility issues that are associated with other preclinical studies. We then perform a replication by identifying the key experiments from those studies, so that allows us to exclude initial exploratory work, and we perform power calculations to make sure that we can detect the effects reported with at least 80% power. We then do rapid and cost-effective replication by using the Science Exchange Network. So this allows us to complete the entire project for $1.3 million. So by doing this project, we've kind of established what we consider to be a high-quality replication plan. So how do people look at replications and make sure that they're doing their best to actually be able to interpret those results accurately? So some of the components that we think are really important for replication studies is that a direct replication is conducted. So unless you use the same materials and methods, it's very difficult to compare the results from a replication versus the original study. We also pre-register the protocols and analysis plan in the Open Science Framework, and this allows us to then obtain input from the original authors so that they can examine the methods and analysis that are going to be conducted up front before data collection. We then use power calculations to ensure that our replication has a sufficient sample size, and use controls to show that the replication experiments worked. We also use expert independent labs from the Science Exchange Network with extensive experience in the techniques being replicated, so that we can ensure that there is technical competence in terms of the experiments that are being conducted. And then finally, we ensure complete transparency by publishing all protocols, results, and data in the Open Science Framework. So the Science Exchange Network is actually made up of labs from across the United States. So there's more than 400 research institutes that participate in the network. And what this is about is creating a way for researchers to order experiments very quickly from technical experts. The goals of the Cancer Biology Reproducibility Project were multiple, so we really wanted to show that direct replication is possible and that it can occur in a cost-effective and scalable manner. We wanted to provide a complete open data set of high-value replicated studies so we can start to do data mining around reproducibility and what things are associated with reproducible studies. We also wanted to develop reproducibility best practices, so we've learned a lot of lessons about how to set up replication studies that we want to share with the research community. And most importantly, we want to create a culture of reproducibility, so we want to demonstrate that reproducibility can be used as an important altmetric to measure research quality. So we've actually just started to extend the cancer biology studies into other fields. So the first field that we'll be extending into is neurodegeneration, and this project is about to launch in the next month. We also want to extend it into multiple other fields once we start to finish the cancer biology studies. So our goal is ultimately to align researchers' incentives. So we really want to encourage reproducibility as an important altmetric, and we believe this should be used to assess the impact of research funding and influence promotion decisions. Thank you.